And I will mostly follow this review paper. It's really nice. It's open access, so you can all download it here. It's a really nice introduction to the topic. It will have more things than actually I will cover today. Again, like normal, if you have any questions, please feel free to raise your hand and interrupt, and we can discuss. So a little bit of history. Of course, you know Galileo Galilei, who in 1610 already started working on microscopy, and these are some of the other. So Antony van Leeuwenhoek is the one that first did already some, biological examples with a microscope. So before I start talking about high-resolution microscopy, we first have to go back to some of the basics of microscopy. And so the concepts, some of the concepts that you all probably know are, of course, refraction. So refraction happens when you go in a medium that has a different refractive index than the other one, you will get the bending of light. And if you do that, this depends on the wavelength of the light. So if you send white light through a prism, for example, which has a different refractive index, then you will get dispersion and you will get splitting of the colors. And of course, what we are going to need today is to talk a little bit about lenses and how we can focus light onto a focal point. So here in this presentation, F will be the focal distance. So another concept that I'm sure you all know is that of fluorescence. So in fluorescence, we have an excitation from the ground state into the excited state or into these fibronic states that relaxes to the first excited state. And then we can get emission, which releases a photon of lower energy than the one you use to excite. So this is the Jablonski diagram. I'm sure you have all seen that before. And if we look a bit closer at all these levels in terms of energies, we see here that there are many levels, and that's why it's not a single energy, but because you have these fibronic levels, you can have actually a broad range of wavelengths that can excite. And often the excitation and the emission wavelengths are a kind of mirror image of each other. So here you have the excitation, and here you have the emission wavelengths. Okay, this is all basics. Another concept that probably you have seen in high school is that of interference. So when I have coherent light and photons that travel alongside each other, they can interfere with one another, and this can be constructive or destructive interference. So if you, at least when I was in high school, we did this experiment with slits. So if you have two slits, each slit will act as if it's, so here you have basically a planar wave of light coming, and each slit will now start acting as a point source that emits light. And where these two beams hit each other, you can have constructive and destructive interference of light. So this is, if you look at the sun through a double slit on a plate, you will see this kind of banded structure. This will be important later when we are going to talk about, for example, confocal microscopy. Okay, so the human eye is of course a very interesting tool because it can actually change the focal distance, right? So if we are looking at an object in the distance, here's the retina, this is the focal point of the lens, and you can do this simple deconstruction of the light path. So here we have the straight one that goes from the lens through the focal point onto the retina, and we can have a small image. So if something is far away, we see it smaller. But if something is closer, actually the focal point of your lens can be adjusted, and therefore you can also see objects that are closer. So in this way, we are not limited to just looking at objects at one fixed distance, but as you know, your eye can adjust in real time. So what is our obsession with resolution? You know, why do we need to get increasingly good resolutions? And that's because things, of course, happen at different length scales. So we, you know, human beings, you know, we are existing on a resolution of 1 meter, roughly. If we look at ants, they are 1 millimeter large, right? If we go even smaller, so the human hair has a diameter of 100 micrometers. If we go even smaller, this is a human cell. Actually, these are 2 cells, and they are roughly of the size of 10 micrometers. If we go even smaller, bacterial cells are another factor 10 smaller, so they're about 1 micrometer. Can go even smaller. Viruses, very, you know, much in the news lately, are on the size of 100 nanometers. These are actually big viruses. Even smaller, this is a single protein. This looks like an antibody on the length scale of 10 nanometers. And nowadays, this is with scanning tunneling microscopy, we can even see single molecules. So this is some kind of coronine family. So these are aromatic rings. And you can see actually a single molecule on a surface with the latest generations of scanning tunneling microscopy. So we're not going to talk about this type of microscopy. We're only going to talk about light microscopy, but it's just to show that many different tools have been developed to look at structures at different length scales. And each one has its pros and cons, the same for the techniques I will show you today. But basically in the last 10, 20 years, there has been a big revolution in the resolution that we can resolve. For example, we're not going to talk about that today, but using, for example, cryo-electron microscopy, people are now solving single protein structures, which before was only done with, for example, X-ray crystallography. So the detectors are becoming super good. The software to analyze those images is becoming increasingly good. And therefore, we're reaching higher and higher resolutions so we can actually see single molecule level events. Okay, so again, the human eye can cover from more or less, you know, a little bit below a millimeter to meters. Magnetic resonance imaging and PET, they are very useful, but not very high resolution. So light microscopy kind of ends at 200-300 nanometers, and we'll talk about why that is. And I'll show you a little bit how we can overcome such resolution issues. And then, of course, there's also electron microscopy, which is for the very smallest things, but for example, not useful for looking at live cells. You know, cryo-electron microscopy is done at cryogenic temperatures, so you cannot have living, you know, structures or beings at those resolutions. Okay, so in the last decade or so, there have been several Nobel Prizes that have been awarded that are related to super resolution techniques. The first one we're going to talk about a lot is the work of Betzig, Hell, and Mörner. So that's basically using different versions of super resolution microscopy. And also, like I said, cryo-electron microscopy has gone through a huge development in the last decades. And so we're not going to talk about it, but it's, of course, very interesting work that is becoming very important at the moment. So the types of microscopy. So again, to repeat, today we're only going to talk about light microscopy, and specifically we're going to talk about fluorescence microscopy. But there are different types of light. So transmitted light microscopy is the simplest one you can think of. So it's just light has a difference in absorption when it goes through a sample. Therefore, you get different amount of transmitted light, so you get some form of contrast. These are more complicated ones. So phase contrast and differential interference contrast are techniques that work on the phase or interference of a sample. So when you look at biological samples like a cell, of course, the difference in absorption is very, very tiny because the sample is very thin. So the absorption is almost zero. But usually there's still a change in the phase of the light, which you can detect with using phase contrast. Or differential interference, you basically split your light into two beams, send them through the sample, and then by interference effects, like I said, for the slit, you can get some contrast. Also, you will get this, I guess, in other courses, is atomic force microscopy. It's a little needle that taps on your surface. You can also get very high resolution and other techniques that we're not going to talk about. So why focus on fluorescence today? Because fluorescence, you can have different colors, right? You can label, for example, if we look at cells, we can look at different types of cells. So we have here red cells and green cells, and we have nuclei in blue. You can have specific staining for specific cellular components. And people have developed also genetically expressed dyes like GFP and YFP that you can encode in the cell, which then express a fluorescent protein in the cell. So you can stain specific cell components. Let's say you want to only stain microtubules, or you want to only stain the Golgi apparatus or some other actin fibers. You can actually genetically do that, or there are specific dyes that bind to these cellular structures. So you can do that at the cellular level, but you can also do this at the subcellular level. So for example, this bright spot here is the centroid. This is where basically microtubules can grow, and we can have different colors for different specific subcellular components in the cell. And these are cool pictures, but the cool thing, of course, is that this can be done in real time. So this is a movie where you're looking at mitosis, so cells dividing, and you see, this mitotic spindle. So you have the centrosomes that are pulling the chromosomes apart. So everything here has been stained. So it's really nice that you can look in real time what is going on in these cellular processes. And that's a very unique factor in using fluorescence microscopy. So it's very heavily used in biology. And the most basic microscope looks like this. So we have here our sample, we have an objective, so this is the lens, and we have different excitation and emissions, right? So we have first the excitation light. So this is the higher power light, higher energy light. It goes through a filter that filters just for a specific band of in the excitation regime. Then it goes on this dichroic mirror. This reflects the excitation light into the objective. Then from the objective, it goes on the sample. The sample will then fluoresce, so it will emit light. And this emitted light is going back into the objective. It goes down, but now, so this dichroic mirror reflects wavelengths below a certain wavelength, and it passes the ones above it. So the emitted light is then going through this dichroic mirror, and it goes into the detector. So in this way, we can excite and observe through the same objective. The light goes up and down through the same objective, and it looks like this. So here we have our sample, and usually this is done either in an inverted or an upright configuration. Now you have microscopes that you can flip around, and those are really cool. But that's basically the basic of fluorescence microscopy. So in the last 20 years, a little bit more, some improvements have been made, and the main breakthrough at that time was confocal microscopy. So what do you do in confocal microscopy is you introduce this dual lens system, but also the most important thing is the pinhole. So what does this pinhole do? If you look at, this is my sample, this is the focal plane, right? So here's where my objective has its focal point. And you can see that the light, the red light, goes through the lens, and it goes actually through this pinhole and then onto the detector. whereas the blue light is from a different focal plane, it will be rejected by the pinhole. So that light doesn't go through the pinhole. So in confocal microscopy, you can look at a very thin slice of your sample. And you think, why is that interesting? Well, two reasons. One is you don't get any background noise from other layers. So if I want to look at the nucleus, I only want to look at the nucleus and not at all the layers that are above. And actually, you can change the focal point and image many layers, and this will give you a three-dimensional image. So with confocal microscopy, I can do three-dimensional imaging of my fluorescent sample, which is really important in the last 20 years. Okay, so a little bit more in detail. So what happens in this pinhole of the microscope? So we have coherent light, so we have laser light that comes from the sample, for example, and that goes through this pinhole. So a couple slides back, I showed you these interference patterns of the sun that give lines. If you go through a pinhole, you don't get lines, but you get this kind of rings. And so this ring is called an Airy disk, because the person that invented that was Mr. Airy. And the way this profile develops is called the point spread function, the PFS. So this is coming. So again, this is the light. It goes through the pinhole, and then by destructive and constructive interference, I get this intensity profile, which is non-uniform. So on the plate, it looks like that. And if you look at it in a cross section, you will have the main intensity peak, and you have smaller fringes around it. So what is limiting the resolution of fluorescence microscopy is that, at some point when there's overlap between two dyes, let's say I have, usually they're the same color, but I have two molecules and they're emitting, it's difficult to separate them if their point spread functions are overlapping. So the distance d, where I can still distinguish the two dyes, and typically that's defined by the fact that this valley between the 2 points should have an intensity drop of about 20% or more. So this value d was predicted by two people, Mr. Abe and Lord Rayleigh. They have different equations, but they're not that different. Basically, it depends on the wavelength of the light, lambda, and it depends on the numerical aperture of the lens that you're using. Right? So there is this what's called the diffraction limit, and we're going to focus mainly on this one. But that basically says, well, for a certain wavelength of light, for a certain numerical aperture, that depends on your lens, you will get a certain maximum resolution, which is the diffraction limit. And there's really no way you can around it. So Ernst Abbe on his memorial has actually the equation etched in there, so about the diffraction limit. And the problem was that because this was such an influential person, and also Lord Rayleigh, of course, was not a stupid person. So no one, people didn't, were not brave enough to kind of go past this resolution. They thought, okay, these are the laws of nature, you cannot beat physics, what they thought. So they didn't really try to overcome this diffraction limit until not so long ago. So now we come into, the different ways of achieving super resolution microscopy. And there are many, and scientists love acronyms. So there are many techniques now that are in use, and all of them are commercially available through different companies like Nikon, Leica, Zeiss, all the microscope builders. But they can basically be divided in three different methods. The first one is structured illumination microscopy. The second is stimulated emission depletion microscopy. And the third one is single molecule localization microscopy. all use very different methods to improve resolution. But these other techniques all fall within those three categories. And so I'll use this abbreviations, so SIM, STET, and SMLM, to indicate the different sections of the presentation. Okay, so the first one is kind of the hardest to understand also. So in structured illumination, we're going to illuminate our sample not just with a normal beam of light, right? So in normal microscope, you have a lamp that shines a uniform bundle of light on your sample. So in structural illumination, we're going to do that differently, and we're going to make use of what's called the Fourier transformation. So if I take this image and I do a Fourier transform, I will get this image. So it's hard to believe maybe that this image actually has the same information content as this. So if I do an inverse Fourier transformation of this image, I will get this one back. So here you see, for example, okay, if I put diagonal lines, I will get this. If I do this kind of overlap of these two patterns, the Fourier transformation will give you this, right? And what's important in the Fourier transformation is that there's, let's say, from the center is where the highest frequency information is stored, and then you go outwards, the frequency changes. So what happens in a microscope? There is a region in the Fourier space that dictates the resolution of information you can capture. So depending on the numerical aperture and the wavelength of the light, I will define a kind of circle, and only in this circle I will be able to record information. Even though out of this circle, probably there's other information that will contribute to resolution that I cannot access because of this law of Mr. Abe, the Abe criterion. Okay, so how can we overcome this limit? Okay, so first some example. So we have this person here, I don't know who it is, and we do a Fourier transform, we get this. If we now say, okay, we have a lens, and this only captures a certain amount of information, we cut out the other part of the Fourier space, and we will transform it back. We end up with a blurry image, which is what happens in microscopy, right? There's a limit to what you can see with microscopy. And you can see that other information is not captured, but if you transform that back, you still have another type of information that we lost because we were imaging with this numerical aperture. Okay, so to understand what's going on, we have to talk a little bit about moire patterns. And moire patterns, you have probably seen them. So if you have little transparent films with a regular pattern, right, and you overlap them, you get the emergence of different types of patterns. And those patterns are usually larger than the ones you initially start with. So you can see, for example, these are, I think, overlapping lines. And if you change the angles, you see this person is changing the angles, you see new patterns emerging, and they are larger, right? So they fall in a different frequency area if you would do a Fourier transform. This is also what happens if you look on TV and someone is wearing a shirt with squares, for example. That's why you should never wear squares on TV, because the TV still has the line scan cameras. Well, now it's changing, but okay, a lot of cameras still have line scans. So the lines of the detector together with those squares in the clothes create these moire patterns that you sometimes see on TV. It's getting less with all this digital stuff, but it still exists. Okay, so these are moire patterns. So how are we going to illuminate our sample with these different illuminations of light? That's done using a grating. So here in this grating, we can see here an image. It's very finely etched piece of glass, basically, or quartz, and you send your laser through that, so you'll get a structured illumination because of this grating, which then goes onto your sample here and into your camera. Okay, so what is happening? This is again the Fourier space, right? I have my zone, which is limited by my numerical aperture and the wavelength of the light. So when you overlap your light in a structured way, so for example, you see in this image, I have a very fine spacing between these two patterns, and you see that bigger lines emerge, right? So what happens effectively is that you can shift the position where you are imaging in your Fourier space, and you can recenter it to the edges of your resolution circle. So what I'm doing is I create a new area in my Fourier space that is now become accessible to image. Then you do this at different angles. So I will make effectively a larger circle that I can image in my Fourier space. So this is an example where they have done that for biological structures. I will show you maybe this image. This is the normal fluorescence microscopy image, and this is after the structured illumination microscopy, where I use my different angles of illumination. And the resolution you gain is about a factor 2. So it's important, but it's not the best resolution you can get. So again, there's a kind of physical limitation that now you've overcome. So you got twice the diffraction limit using structured illumination. If you have questions, please feel free to interrupt. This is another image. So here again, this is normal microscopy. Let's say this is already confocal microscopy. And then this is SIM microscopy. So you see there's a clear improvement in the resolution. So what are the pros and cons? So the pro is it's simple. You don't need any special fluorophores or dyes, and it can be done quite fast, which if you are looking at live cell imaging, that's actually an important limit. But the resolution is only improved twice, and it can have a limited sensitivity of what you're looking at. Okay, so the second technique is called stimulated emission depletion microscopy, or STED. And one of the important people that developed this technique is Stefan Hell. And you see that he actually has his own equation, which looks kind of like the Abbe equation. So you have the wavelength, you have the 2n sine alpha, and you have this new term here where you have J and J saturation. So we'll come back to what that means. Okay, so what is this method using? It's using stimulated emission. So what does this mean? If you have, basically you can get excitation, right? So you excite to the excited state, and then you can get emission. But if you excite with a really high power, you can actually deplete photons from the excited state, and you can make those states darker. Okay, so how does that work? So that is using something called the depletion donut, and it's called donut because it looks like a donut. So I'm going to excite with a very narrow beam of light, and I'm going to deplete. So I'm going to turn off the fluorescence using this donut beam. So if I put them on top of each other, I will get fluorescence from a bigger part, from this beam. But the depletion beam is going to make everything around it dark. And the result is that I'm going to get an effective point of light, which is way smaller than what you could normally do with a normal beam of light. Okay, so what does that look like? So we have the excitation, we have the depletion. Together you get this stimulated emission, which means the fluorescence will be very, very, very narrow from only this spot. Okay, so this depletion, it's done immediately after the excitation. So we have excitation, and then so this depletion pulse is some picoseconds after the excitation. So we excite all the molecules, the depletion beam comes and turns off the fluorescence around the spot, and I will get some nanoseconds later, I will get fluorescence that will be then detected again. So it's a pulsed technique. And we have a little animation to look at that. So we're looking at first at a normal confocal beam. So you see the lens, and at the focal point, the beam will be very narrow. And we have two fluorophores, the red dots. And we're going to scan our beam through our sample. And when we excite, the dyes will emit, and you get a profile like that. So we only see one peak because the molecules are too close to each other to be differentiated because of diffraction limit. But here, if we have the depletion beam around, you see that the spot size is actually much narrower. So we are able to distinguish the two dyes, even though they are very, very close to each other. Okay, another interesting point, coming back to this equation, was that there was this factor square root of 1 plus J over Js. So that's the power of the saturation beam. So actually instead the resolution, so this is the normal diffraction limit, which is about 200 nanometers. But if I increase the power, I actually also increase the resolution. So I can go down to 50 nanometers because my depletion beam is getting more intense, and therefore the point where the excitation occurs gets smaller and smaller. So the power you need in this case, so this is again the equation, is this the intensity of the saturating beam of the depletion donut. So we have, this is the wavelength of the light, this is the numerical aperture. So all the other factors are fixed. We can only play with this saturation beam. So just for your information, like a confocal microscope uses about kilowatt intensity. So already you see these numbers. It's 3 orders of magnitude stronger than confocal microscopy. So you need a quite brutal laser to get this depletion beam. And also, of course, you only use it for very short times. So if you look, lasers can be very intense and they can actually cut things. So you need very high powers. And that's kind of what's limiting this resolution now. If you want to go to 5 nanometers, you need 20 gigawatts of power, which is absolutely insane. So that's what's limiting the resolution of STAT at the moment. But you can get some very beautiful pictures. So here we have confocal microscopy images. This scale bar is 200 nanometers, and you see that with STAT, you have a much higher resolving power as compared to the normal confocal microscope. Also, this technique has now been used in supramolecular chemistry, for example. So here you have an example where you get supramolecular fibers and they self-sort, which means they do not co-assemble between green fibers and red fibers. And using quite standard dyes, we can now image these fibers. So here you see 3-dimensional imaging, again, of the, let's say, normal way of imaging versus the super resolution technique. And you can even do that in 3 dimensions. So STAT is just like confocal microscopy. You can also image in 3D, but at much higher resolution. So we can really start seeing, well, this phenomenon would only be visible, with very high resolution imaging. Otherwise, you would just see a combination of those two colors together. Okay, so this was stimulated emission depletion microscopy. So you have very good resolution, 30 to 50 nanometers. It's simple to use. It's easy to penetrate into the sample. You can do it on live cells, right? So the cells can still be alive. You don't have to fix and stain them. It's moderately fast, but it's very expensive. It's difficult to maintain and to calibrate. You need high power lasers, and you need special dyes that are actually undergoing this stimulated emission, right? So not all dyes that are commonly used in chemistry or in biology can be used for this microscopy technique. And so this is Stefan Hell in the middle. This is John Pendry, and this is Thomas Ebbison. So who, the three of them together were awarded the Kavli Prize. So it's after the Nobel Prize. The Kavli Prize is the most important in nanotechnology. Thomas Ebbison, of course, not for super resolution microscopy, but for extraordinary light propagation through small, tiny holes of light. But this was an important event. Okay, is it clear? I'm going too fast? No? Okay. Okay, so the last technique I want to talk about is single molecule localization microscopy. And this is using, this is a completely different way of imaging. Right, so what I'm going to use is I have some object, and if you do WF is wide field imaging, I would see all these fluorophores together. So I won't be able to distinguish them because of the diffraction limit. So in single molecule localization microscopy, what I'm going to do is I'm going to take many images. And I'm going to make sure that in each image, not all the dyes are showing up at the same time. We'll show you how that works. But if only a few molecules are lighting up, I can actually determine their location much more accurately than I would be if I have this issue with the diffraction limit. So I'm taking many images, and in each image, different dyes are blinking. But since they are well separated, I can localize their center by fitting the point spread function onto that profile. And therefore, I can determine their location. And then I can sum that up, and I can actually get very high resolution images of my object. Okay, so this is going to be a movie of what's happening. So you're going to look at a sample. I think it's a cell. And you're going to see this kind of flashing of lights, which are the individual dyes that are fluorescing, but stochastically. Right? So you see all kind of blinking spots of light, and each blink will allow the algorithm to fit the exact position of the dye. And we put that on top of the image. So this is, whereas in the movie, you saw this hugely pixelated blinking image, after reconstruction, we can get very high resolution image because we are only seeing a few dyes at the same time, and we're not looking at everything at the same time. Okay, so how precise can we get? So here we have the localization precision. So this is the distance. It depends on the number of photons that you collect on your detector, on the standard deviation of the point spread function, so how wide your point spread function is. Of course, on the pixel size of your detector, if you get a more a detector with smaller pixels, you will be more accurate in finding the location. And also on the background noise of the pixel. So a lot of detectors, so their cost goes up because the dark count, as it's called. So if you don't have light on your detector, the detector sometimes still will give a small signal. So you want to have a detector which has a low dark count, as it's called. But they exist, right? So that's okay. So that depends on your detector, on your light source. What other things can you do? So when you look at small molecule dyes, so this is an organic dye. This is some kind of photo-caged rhodamine derivative. It's small, right? 1 nanometer. But if you go to proteins, and a lot of detection in biology is done by antibodies. And often, so if you have, for example, an ELISA test in biology, you have a first antibody, and then you have a second antibody, which binds to the first antibody, and that antibody has a fluorescent protein. So if you're looking at such a construct to stain a specific part in the cell, for example, this whole thing together is already 20 nanometers. So we're never going to be able to get high resolution if my whole construct to make it fluorescent is already so huge. So that's why, for this kind of imaging, the highest resolution is obtained by using fluorescent dyes. Another issue is that, for example, your sample can drift. We're looking now at, look at the scale bar, we're looking at nanometer sizes. So any, temperature fluctuation in your room, in your sample, will create a small thermal drift in your image. So what you can do for that is mathematically you can track each spot. And so if you do a drift correction, you just say, okay, I have this spot, which is always staying in the same place, and I'm going to correct all my images for these thermal fluctuations. And that increases your resolution a lot. Okay, so you can add, for example, a very strong fluorescent particle that doesn't blink, that doesn't photo bleach to your sample, which allows you to correct for the drift in your sample. So then there are different methods for getting this blinking, right? So we want to get a dye that has either 2 states or that is photo bleached. So in this first technique, which is called stochastic optical reconstruction microscopy, or STORM. So in STORM, actually we are going to use photoredox switching. So we have a molecule which is fluorescent. We shine light, we get a reduction to the reduced state. And in this case, the reduced state is non-fluorescent. But this is reversible. So we can, using a different wavelength, oxidize the dye back to the fluorescent state. So we get an on-off switching of this dye. So it's reversible. How can that be done? For example, in this molecule, we have here the fluorescent molecule, which can be localized. And using this red light illumination, we have a tile that reacts here with this double bond. And this is actually a dark state. Oxidation with blue light removes the tile again, and you get back the fluorescent molecule. So in this way, we are photochemically turning on and off the emission of the light, which leads to blinking, which allows us to actually find the exact location of that dye molecule. The second method is called photoactivation localization microscopy, PALM. And actually that uses irreversible photoactivation. So we have a dark state, we shine some light that triggers fluorescence. and then this fluorescence will disappear by photobleaching. So you know that almost any molecule fluorescent, if you put it in a strong intensity light, it will break. You get what's called photobleaching. It happens for almost every system you can imagine. So photobleaching causes the intensity of the dye to increase versus time, right? So if you have intensity, so in a photobleaching process, you have an exponential decay of the fluorescence intensity over time. Okay, so let's see what that looks like. So we have our initial view. We have a laser beam which activates some of the dyes. So these ones are activated. Because they are separated more than the diffraction limit, we can localize them, and they photobleach, right? So they disappear over time, and then we can activate again on the same area. We shine the same beam again, but now we activate 3 new molecules. We localize them, they photobleach, shine the beam again, localize, photobleach. So it's a combination of photochemical activation and photobleaching that results in the blinking of the dyes. So for example, here you have, this is a photocaged fluorescein, so it's dark. When we shine light, we lose those nitrobenzene groups, and you get fluorescein, which is fluorescent. And if we shine enough light, of course, this will bleach and it will disappear. So all of these are different kinds of dyes that are activated. So in this image, you can see here, we're going to shine some UV light that triggers fluorescence. And then that results in bleaching and blinking. So there was palm. Yet another way is using paint. So that's called point accumulation imaging in nanotopography. So in paint, the dye itself is not chemically changing or being chemically activating, but it does change in fluorescence depending on its environment. So for example, we have a structure that we want to image, and we have a lot of these dyes in solution. And only when the dye binds to that structure, it becomes fluorescent. So a very common dye that does this is, for example, Nile red or thioflavin T. Those are dyes that they change their emission depending on the hydrophobicity of their environment. So if they bind to a hydrophobic membrane, for example, they will become much more fluorescent, and they will bind and unbind. So it's the binding and unbinding that creates the blinking of the dye, and therefore the ability to localize those structures. So for example, here we have some fibers. Here you see the diffraction limited image. And in this case, there's on top of these fibers, there is an antibody that binds to a strand of DNA with a fluorophore. And this fluorophore only becomes really fluorescent when it's in proximity of this antibody. So only when it binds, it becomes fluorescent, then it unbinds, you don't see it anymore. And then we collect for a long time, we can actually see this much higher resolution fibers in the image. Okay, so with this poster is a little bit small on your screen here, but on the slides, which I will put on Moodle, you will be able to see it better. So if we go through the different steps, so we have normal fluorescence imaging, which is limited by the diffraction limit. So about 200 nanometers is the smallest you can see. Then we talked about structured illumination methods. So it's overlaying patterns of intensity of excitation light onto your sample, rotating that many times, and then mathematically deconvoluting what is actually underneath. So this is giving you about a factor 2 in resolution. Then we talked about stimulation depletion microscopy. So we have the excitation beam and the depletion beam together, giving a much smaller spot where the light is coming from, therefore going to a resolution of about 30 to 70 nanometers. And then we have the single molecule localization method, so PALM, STORM, resolved, that actually give you resolutions that are much below the diffraction limit. So even 10 nanometers is now possible. So we're already 20 times below the diffraction limit. Okay, with that, I want to take a small break, maybe 10 minutes, and then we continue.